We'll demystify CRISPR-Cas9, how a bacterial immune system became a precise genome editing tool, the molecular steps that cut and rewrite DNA, key applications from medicine to agriculture, and the technical and ethical challenges shaping its future. Chapter 1: Origins and Molecular Background. CRISPR loci were first noticed in bacterial genomes in 1987 and recognized as an adaptive immune system by the mid-2000s. A CRISPR array contains short direct repeats interleaved with variable spacers, molecular memory sequences derived from phage or plasmid DNA. When a bacterium re-encounters the same invader, CRISPR RNAs, cRNAs, guide Cas nucleases to complementary sequences and trigger degradation. Cas proteins are diverse. Cas9 from Streptococcus pyogenes became the workhorse for genome editing because a single protein both recognizes DNA and makes a double-strand break. Effective targeting requires a protospacer adjacent motif, PAM. For SP-Cas9, this is typically NGG, a critical 2 or 3 base signature next to the target. Chapter 2: How CRISPR-Cas9 actually works. At the molecular level, Cas9 is directed by a 2-part RNA, a CRISPR RNA, cRNA, that matches the target sequence, and a trans-activating cRNA, tracker RNA. In engineered systems, these are fused into a single guide RNA, sgRNA. Cas9 first recognizes the PAM, unwinds nearby DNA, and forms an RNA-DNA hybrid. Cleavage occurs approximately 3 nucleotides upstream of the PAM, producing a double-strand break, DSB. Cells repair DSBs by non-homologous end joining, NHEJ, which often introduces small insertions or deletions, or by homology-directed repair, HDR, using a template. HDR efficiencies vary widely, often below 20% in many somatic cells. Newer approaches, base editors and prime editors, make precise changes without creating blunt DSBs, increasing safety and precision. Chapter 3: Practical Applications and Real-World Examples. CRISPR has rapidly moved from bench to real-world tests. In medicine, ex vivo editing of hematopoietic stem cells is in clinical trials for sickle cell disease and beta thalassemia, with several patients experiencing substantial symptom reduction after autologous infusion. CRISPR-based diagnostics, for example, Detector and Sherlock, can identify viral or bacterial nucleic acids in under an hour, enabling rapid point-of-care tests. Agriculture benefits include crops engineered for disease resistance or reduced browning. The USDA in 2016 treated a CRISPR-edited non-browning mushroom as not subject to regulation. In ecology, laboratory gene-drive constructs in mosquitoes have demonstrated greater than 90% biased inheritance, illustrating powerful but risky population-level interventions. Chapter 4: Limitations, Safety, and Ethical Implications. Despite promise, CRISPR faces technical and ethical constraints. Off-target cuts and on-target mosaicism remain concerns. Improved enzymes such as SP-Cas9 HF1 and ESP-Cas9 reduce unintended edits but do not eliminate risk. Delivery is a major hurdle. ADNO-associated virus vectors have a number 4.7 kb payload limit, prompting use of smaller nucleases, e.g. Cas9, or non-viral delivery like lipid nanoparticles and ribonucleoprotein electroporation. HDR is inefficient in non-dividing cells, limiting some therapies. Ethically, germline editing provoked global condemnation after heritable edits were reported in 2018. Many countries prohibit clinical germline modification. Governance, equitable access, and robust safety testing will shape responsible deployment. CRISPR-Cas9 rewired gene editing by converting a bacterial immune mechanism into a programmable DNA cutter. Understanding PAMs, guide RNAs, and cellular repair pathways explains both its power and limits. Continued advances in precision, delivery, and governance will determine whether CRISPR fulfills its therapeutic, agricultural, and scientific promise responsibly.